In this video, we're going to take a look at photosynthesis. So for photosynthesis, we kind of have this mystery. Plants don't eat, but yet they grow. And so, of course, we know that plants don't eat. We don't see them with mouths or doing any of those traditional things. And this was something that in the mid-1600s, John Baptista van Helmont did an experiment. He put a little willow shoot, so a little baby willow, into a bucket of soil. And he wanted to know how this was growing. So he was looking at, how the willow was changing its weight and how the bucket of soil was changing its weight, trying to figure out where this mass was coming from. So after he planted the willow, 5 years later, he weighed the tree, and then he weighed the soil. So he's, carefully separated and weighed each of them. He found that the tree had gained 164 pounds, but the soil had only decreased by 2 ounces. So he proposed this question: where did the increased mass of the tree come from? Where are the molecules that are making the tree bigger coming from? And I want you to take a minute and kind of think about that. And now if you think about that, when you have a plant growing in a pot, the one thing you're doing is watering it. And so as you add water, that's adding hydrogen atoms, and oxygen atoms. But when you're adding to the mass of a tree, what are you actually adding to? What kind of molecules are you adding into the tree? So in order to grow a tree, you have to grow cells. And in order to make more cells, what are the molecules that compose a cell? This is where we began this whole discussion in this course. We said, biomolecules are going to be fats and sugars and proteins and nucleic acids. And all of those molecules contain carbon. But Jean-Baptiste van Helmont was only adding water to the pot. So where was all of this mass, this 164 pounds, I guess minus those 2 ounces, but where was this 164 ounces coming from? It could not be explained just from the water, because the things that were adding mass to the tree were carbon molecules, and we know that that's what composes a cell. So the question he asked is, where did the carbon molecules that compose the tree come from? And this is the mystery that we're going to solve as we look at photosynthesis. So kind of keep this question in mind as we're moving forward. Okay, so for photosynthesis, this is going to look a little bit like what we just talked about with cellular respiration, as far as we're going to be forming glucose, C6H12O6. This is glucose. Now, in this little formula here, I have just CH2O. We could write that as CnH2nOn, where if we sub n equals 6, we're going to end up with C6H12O6, which is glucose. So this is just kind of a generic formula that is showing us how we synthesize glucose. And if you look at this carefully, this reaction looks a lot like cellular respiration, except for cellular respiration went this way. And for cellular respiration, it was glucose plus oxygen. And we went this way, and we produced water as a waste product and carbon dioxide as a waste product. Well, with photosynthesis, we're going the other way, and we're going to be producing glucose as a product, and then oxygen will be the waste product. Now, also note, this delta G is the same value that we saw with the breakdown of glucose, except for the breakdown of glucose, that delta G was a negative 685 kilocalories per mole. And because now we're doing it in the opposite direction, so it was negative when we were going this way to break it apart, so as it was energy releasing, that delta G was a negative number because that amount of energy was being released as an exergonic reaction. Now that we're going this way, this is a positive number because this is an endergonic or energy requiring reaction. And so kind of one of the questions we're going to ask is, what is this exergonic reaction, or where is this energy coming from in order to enable us to synthesize this glucose molecule? In this equation, just to kind of clarify other things that we've done with the A here, the A here, the A, what we need to follow really if we want to look at traditional photosynthesis is oxygen. So you would just sub in here an H2O and an oxygen here for that A. But the reason that we wrote it as an A instead of putting in an oxygen is because it could also be sulfur. And so that would be to make hydrogen sulfide and to make sulfuric gas would be another option for how this could run. But so when we're looking at photosynthesis, another way of looking at it is, once again, we're going to have something is reduced and something else is oxidized. So this is kind of a great big oxidation reduction reaction. And so when we're thinking about what is reduced, to be reduced means to gain energy or electrons, and you are reduced. So here our carbon dioxide is going to be reduced. So as it becomes glucose, we are reducing it because it is gaining energy. That glucose molecule has more energy, and it's gaining electrons. And so if you follow CO2 to CH2O or glucose as it would be, we have to put a 6 here. 6 carbon dioxide molecules will get us to this 6 carbons in glucose. But if you're following what's happening here, really it looks like what was gained is the H. And that's because, if you remember, a hydrogen atom has got the proton in the middle and the electron going around the outside. So when we say we're gaining this hydrogen, well, we're also gaining that electron. And so we say we gain electrons to be reduced. You look at it and you say, wait, I don't see how you gained an electron because you gained a hydrogen. Keep in mind that that hydrogen is including an electron. So when you say it gains that hydrogen, sometimes I just follow that hydrogen for this process of reduction or reducing compounds. Okay, so if something is reduced, something else has to be oxidized. It's an oxidation reduction is always coupled. And so in this reaction, we're seeing water is going to be oxidized. So to be oxidized means you lose electrons and you're oxidized. So water, as it becomes oxygen, O2, it's losing those electrons. And if you look at it again, you go, oh, wait, well, it didn't lose electrons, it lost hydrogen. Well, that's because if you lose hydrogen, an electron's going with it. So a lot of times when we follow oxidation reduction reactions, we talk about following the energy and the electron, but then what you see is frequently going to be that hydrogen in a lot of these oxidation reduction reactions. Okay, so as we look at things that are able to do photosynthesis, we sometimes call them carbon fixers, because what they're doing is they're fixing these carbon dioxide molecules into a glucose molecule. So they're fixing it. So we call them carbon fixers. And the kind of organisms that are able to do this are called autotrophs. So autotrophs are going to be able to do this process of photosynthesis, or this process of carbon fixing, in order to take CO2 and turn it into a glucose molecule. Autotrophs are able to do this. They produce, another way of saying that is they're producing organic molecules from an inorganic substance, because this carbon dioxide is considered inorganic because it's just carbon dioxide gas. And then glucose is one of our biomolecules, so that's one of our organic molecules. And so autotrophs, another way of seeing what this word is saying is it's self-eating, meaning they make their food, and then they make glucose, they fix glucose, so this is their food. And then they also do cellular respiration, just as we were talking about last week, in order to use that stored energy from that glucose to run a lot of other processes in the cell. So the thing about an autotroph is it makes its own food, and then of course it eats its own food. And so that's going to be anything that can do photosynthesis, the green plants, the algae, and even some types of bacteria. Everything else then on this planet is technically a heterotroph, meaning it eats other things. And so we're kind of seeing this with this whole food chain. Most of the things here are considered heterotrophs. They eat other things. And so we actually get all of our molecules from the autotrophs. And so here we're trying to understand, how do the autotrophs do this? How does this process of photosynthesis, which everything else in the food chain relies on, how does it work? So photosynthesis is this process of producing organic molecules. So that's defined as any molecule that contains carbon, with the notable exception of not carbon dioxide. So that's actually part of the definition of an organic molecule. An organic molecule is any molecule that contains carbon, with the exception of CO2. So that's how that kind of works out. CO2 is considered inorganic. So this is an inorganic source, water and CO2. So for photosynthesis, we take these inorganic sources, water and CO2, and we put them together to make an organic molecule. So what we're trying to answer in this video is, or in the next series of videos, is how is it done? How do we do photosynthesis? And kind of our sneak peek is we're going to take energy from the sun, we're going to capture it, and we're going to use that energy. So this is our energy source. It all comes from the sun. Great big nuclear reactor in the sky provides energy for all life forms on this planet, autotrophs, and then the heterotrophs that eat the autotrophs. So the energy in this process of photosynthesis, we have to first capture the light and then use that energy from the light in order to synthesize these glucose molecules. And another way of saying that is to fix the carbon from the carbon dioxide into glucose. And so this is going to have two stages. It's going to have what's called the photo stage, which is the light reactions. And then it's going to have the synthesis stage, which is going to include the Calvin cycle. And so we'll look at each of these individually. All right, so let's go back and review what the chloroplast looks like. So the chloroplast was a bit like mitochondria. So the mitochondria was featured for cellular respiration. The chloroplasts are going to be featured in this process of photosynthesis. So both of these were considered the semi-autonomous organelles. We had talked about them previously as far as maybe they were originally prokaryotic species that got engulfed by one of the very, very early eukaryotic cells, and then they adapted and grew together over time. To give some of these bigger cells these special features, for example, being able to do photosynthesis. So the chloroplast is kind of like the mitochondria in that they're semi-autonomous organelles. They're also similar because they have both an outer and an inner membrane, which gives them that intermembrane space, or the space between it. Now, unlike the mitochondria, the inner membrane here is also smooth. So these two outer and inner membranes are roughly parallel, and then you have this intermembrane space in between them. In addition, though, the chloroplasts are going to have a third membrane system called the thylakoid membrane. And so this third membrane system is seen here in the middle. These are these green stacks. They're called thylakoids, but they have this outer membrane around them, which has all of this additional surface area. So this is called the thylakoid membrane. And in this picture, the thylakoids appear green because they actually contain a green pigment, which is a chlorophyll you may have heard of. But because they have these thylakoid membranes, they also have what is called the thylakoid lumen, which is going to be the space inside the thylakoid. So that means that we have a lot of spaces and a lot of membranes here. So we have the thylakoid membrane itself. Then we have this lumen space inside the thylakoid, so the space inside here. We also have the space outside the thylakoid lumen, but inside that inner membrane. That's called the stroma. And then we have the inner membrane, and then we have the space between the inner and outer membranes, which is called that intermembrane space. And so we're going to see where the photo part happens, the light reactions, and we're going to see where the Calvin cycle, the second part, the syntheses reactions happens for photosynthesis. The light reactions are actually going to happen on this thylakoid membrane. And I kind of remember that because the thylakoid membrane is green, and we're going to see in the next video that the color is because of the light. So the fact that they're green is because they are absorbing and reflecting some of the specific wavelengths of light. So that's why they appear green. So the light reactions happen on this thylakoid membrane. That's why these are pigmented membranes, because they contain pigment molecules. And we'll see more about that in the next video. The synthesis part, the Calvin cycle, happens in the stroma. So it happens in the space between the thylakoids. And so this space is full of fluid, but we're going to see that there's going to be lots of enzymes and molecules moving about here for what's called the Calvin cycle, which might appear a little bit like the TCA cycle or the citric acid cycle, but of course maybe we could define it as being completely opposite of the citric acid cycle. Not completely opposite, but we'll see that it's fairly opposite of that. And so that's happening out here in this space. Okay, and then all of this happens within the leaves of plants. So leaves of plants are really cool to look at because if you look at them under a microscope, you can clearly see the cells, the plant cells, and inside the plant cells, which are relatively rigid because they have these firm cell walls. Inside the plant cell, you can see all of these little green dots, which, as you might imagine, are the chloroplasts, and you can actually see them moving around inside the cell, which is really cool to watch. And so where all these cells containing the chloroplasts are found is usually on the inner part of the leaf called the mesophyll. So the outer part of the leaf has this epidermal layer, just this barrier layer of cells kind of tightly packed together. But in the middle part of the leaf, there's going to be all of these cells that contain lots and lots of chloroplasts. And if we kind of zoom in on what one of those cells look like, it looks like a typical cell with its nucleus and all the other things, but then it's going to have lots of these chloroplasts kind of floating about it as well. And so that gets us to the picture of the chloroplast here with those thylakoid membranes, the stroma space, and then both an inner and an outer membrane that we saw previously. One last thing to kind of note is we're going to see with the chloroplast that carbon dioxide is going to be used, so that needs to go in, and then oxygen needs to come out. So we have this respiration or this gas exchange, and that happens through holes called stomata, which allow these molecules to move in and out. We can kind of see a little stoma here where you would allow water, these gases to go in and out of the leaf. All right, so that kind of sets us up for what we're going to see with photosynthesis. Just to go back real quick, look at this question again. So if plants aren't eating, how are they growing? How are they getting increased mass? So how are they making the biomolecules that make themselves bigger? And so the biomolecules are things like fats that are going to be in the lipid membranes. As they make more cells, they're going to need to make more fats to make the lipid membranes. They're going to need more amino acids in order to make the proteins to give the cells different kinds of functions. As you duplicate cells, you're going to need more nucleic acids, and you're also going to be using or creating glucose to increase the mass of cells, or this would just be carbs, we could say. And so the question is, how are we getting all of these biomolecules to increase the mass of the plant? And now that we've kind of a little bit looked at it and looked at this reaction of carbon dioxide plus water, this is our answer, right? So, we said previously, we know that we're watering the plant, but that wasn't enough to explain how the plant grew its mass. And so, John Baptista is looking at this and he's scratching his head and he says, could the plant simply be increasing its mass out of thin air? And I think that we would all answer, yes. The carbon dioxide in the air becomes these biomolecules that are the mass of the plant as we go through this process of photosynthesis. So we'll look at this in more detail on the next video.